Welcome to Fall Asleep Channel. Tonight, how chronic stress gradually reshapes the brain, hormones, and body over time. Now allow yourself to get comfortable and enjoy quiet and pleasant rest. Let's begin. Chronic stress does not begin with intensity. It begins with repetition. A signal that was designed to be brief remains active longer than necessary. The body's stress response evolved to respond to immediate threat. Heart rate increases, breathing accelerates, muscles tighten, glucose is released into the bloodstream. These changes are efficient when short-lived. They prepare the organism for action. Yet when activation persists, the same mechanisms begin to alter baseline physiology. The process often starts in the brain. Sensory information is interpreted as significant. The amygdala evaluates emotional relevance. If threat is perceived, it signals the hypothalamus. The hypothalamus activates the sympathetic nervous system within seconds. Adrenaline is released from the adrenal medulla. Heart rate rises. Blood vessels constrict in some regions and dilate in others. Blood flow shifts toward large muscle groups. Digestion slows because energy is redirected. If the stressor continues, the hypothalamic pituitary adrenal axis becomes involved. The hypothalamus releases corticotropin-releasing hormone. This stimulates the pituitary gland to secrete adrenocorticotropic hormone, which travels through the bloodstream to the adrenal cortex. Cortisol is released. Cortisol supports prolonged energy availability. It increases glucose production in the liver. It influences fat metabolism. It modulates immune activity. Under normal conditions, cortisol follows a daily rhythm. Levels rise in the early morning, gradually decline toward evening. This rhythm supports wakefulness and recovery. During chronic stress, this pattern may become flattened. Levels remain elevated when they would normally fall, or become dysregulated in timing. Such disruption affects sleep onset and depth because cortisol and melatonin operate in opposing cycles. Persistent cortisol exposure influences the hippocampus, a region involved in memory formation and contextual regulation of stress responses. High cortisol over extended periods may reduce dendritic branching within hippocampal neurons, affecting memory consolidation. At the same time, the amygdala may become more reactive, reinforcing vigilance. This shift alters how future stimuli are interpreted, increasing sensitivity to perceived threat. The prefrontal cortex, responsible for executive function and impulse regulation, may experience reduced activity under prolonged stress. Decision-making becomes less flexible. Attention narrows. Working memory capacity decreases. These changes are adaptive in acute danger, but inefficient when sustained over months or years. The cardiovascular system responds continuously to sympathetic activation. Heart rate variability decreases, indicating reduced parasympathetic influence. Blood pressure may remain elevated. Vascular tone becomes less responsive to relaxation signals. Over time, this sustained strain increases risk for structural changes in blood vessel walls. The immune system also shifts under chronic stress. Cortisol initially suppresses inflammatory responses, yet prolonged dysregulation may produce paradoxical patterns. Low-grade inflammation can persist. Immune cells alter their sensitivity to hormonal signals. Wound healing slows. Susceptibility to infection may increase. Muscle tension remains subtly elevated during ongoing stress, especially in the neck, shoulders, and jaw. Sustained contraction reduces local blood flow, contributing to discomfort and fatigue. The body maintains readiness even in the absence of immediate action. Digestive processes are similarly affected. Sympathetic dominance reduces motility and secretion. Appetite regulation shifts. Some individuals experience decreased hunger, others increased intake driven by cortisol's influence on reward pathways. Microbiome composition may change when gut motility and immune balance shift, linking stress physiology to digestive function. Sleep architecture often becomes fragmented. Sympathetic arousal delays the transition into deeper stages. Rapid eye movement cycles may shorten. Nighttime awakenings increase because vigilant systems remain partially active. Without sufficient restorative sleep, cortisol regulation becomes further impaired, creating a reinforcing loop. Metabolic patterns adjust as well. Chronic elevation of glucose mobilization can alter insulin sensitivity. Fat distribution may change, particularly with increased abdominal storage influenced by cortisol. Energy that was meant for immediate survival becomes chronically available, yet not always expended. The parasympathetic nervous system, responsible for rest and recovery, becomes less dominant when stress persists. Vagal tone decreases. Respiratory rhythm may remain shallow. Digestion remains subdued. This imbalance shifts baseline physiology away from restoration. Neurotransmitter systems adapt gradually. Prolonged stress can influence serotonin and dopamine signaling, altering mood stability and reward perception. Motivation may decline. Pleasure responses may diminish. These shifts reflect biochemical adaptation rather than sudden change. Over extended periods, the concept of allostatic load becomes relevant. Allostasis describes the process by which the body maintains stability through change. When adaptive systems are repeatedly activated without adequate recovery, the cumulative burden increases. Wear and tear on regulatory networks accumulates, not as visible damage, but as altered thresholds and reduced flexibility. Yet the system remains dynamic. Stress pathways are not permanently fixed. Neuroplasticity allows structural and functional adjustment. Cortisol receptors regulate their own sensitivity through feedback mechanisms. Inflammatory markers fluctuate in response to environmental conditions. Recovery is possible because regulation remains responsive. Chronic stress, therefore, represents prolonged activation of mechanisms designed for short-term adaptation. The same hormones, neural circuits, and autonomic responses that protect in acute situations gradually reshape baseline function when engaged without sufficient resolution. Heart rate, immune signaling, metabolism, cognition, and sleep all adjust to a new equilibrium, one that prioritizes vigilance over restoration. Beneath daily awareness, these processes operate through measurable biochemical pathways. Neurotransmitters released, receptors activated, genes expressed, proteins synthesized, and feedback loops recalibrated. The body does not distinguish between psychological and physical threat at the level of stress circuitry. It responds to perceived significance. And so chronic stress unfolds not as a single event, but as sustained activation across interconnected systems. endocrine, neural, cardiovascular, immune, and metabolic, each adjusting incrementally to repeated signaling, maintaining function while gradually shifting balance, until recovery mechanisms regain influence and regulatory rhythms slowly re-establish steadier patterns across the organism. As prolonged activation continues, subtle structural adaptations appear within neural circuits. Synaptic connections reorganize in response to repeated signaling. Neurons that fire together strengthen their pathways. Vigilance-related networks become more efficient, while circuits supporting relaxation receive less reinforcement. This shift does not occur abruptly. It develops gradually through repetition. The amygdala may increase in functional connectivity with other limbic regions, enhancing rapid detection of potential threat. At the same time, communication between the prefrontal cortex and limbic structures may weaken under sustained cortisol exposure. Regulatory signals that normally dampen reactivity become less effective. This alters threshold sensitivity, making smaller stimuli capable of triggering a stress cascade. Within the endocrine system, feedback loops attempt constant recalibration. Cortisol normally inhibits its own production through negative feedback at the hypothalamus and pituitary. Yet prolonged elevation can reduce receptor sensitivity, diminishing the efficiency of this braking mechanism. The signal to stop becomes quieter, even while activation persists. The autonomic nervous system also adjusts baseline tone. Sympathetic activity may remain slightly elevated even at rest. Parasympathetic recovery responses may initiate more slowly. Heart rate variability reflects this shift. Reduced variability indicates less flexibility in cardiovascular regulation. Flexibility is central to resilience because rapid adjustment between states supports efficient adaptation. Chronic stress influences gene expression through epigenetic mechanisms. Methylation patterns may change in response to hormonal exposure, altering how certain stress-related genes are transcribed. These changes do not modify DNA sequence, but they influence how strongly genes are expressed, linking environment and physiology through molecular pathways. The inflammatory response becomes particularly complex under prolonged stress. Acute cortisol release suppresses inflammation, yet chronic dysregulation may produce immune cells that become less responsive to cortisol signal. Inflammatory mediators such as cytokines may remain slightly elevated. Low-grade inflammation can influence mood, energy levels, and vascular health. In the gastrointestinal system, sustained autonomic imbalance alters barrier function. Stress hormones influence tight junction proteins within the intestinal lining. Permeability may increase subtly. Immune signaling within gut-associated lymphoid tissue adjusts accordingly. Microbial populations respond to changes in motility and secretions, demonstrating how stress integrates neural and digestive systems. Energy metabolism adapts to repeated demand for glucose mobilization. The liver continues gluconeogenesis under cortisol influence. Muscle tissue may alter insulin sensitivity. Adipose tissue distribution shifts in response to endocrine signaling. These changes represent attempts to maintain readiness, yet over time may influence metabolic efficiency. Sleep disruption further modifies stress physiology. Reduced deep sleep impairs growth hormone release, which normally supports tissue repair. Slow-wave sleep contributes to glymphatic clearance within the brain. When fragmented, Metabolic byproducts may accumulate more slowly, reinforcing cognitive fatigue. Circadian rhythm disturbances alter the timing of cortisol peaks, complicating hormonal balance. Musculoskeletal patterns remain subtly engaged. Posture may shift toward protective positioning, shoulders elevated, jaw muscles active, diaphragm movement shallower. Chronic contraction alters proprioceptive feedback, influencing perceived tension even when external stressors subside. The cardiovascular system continues adapting to sustained sympathetic tone. Endothelial function may change in response to repeated vasoconstriction. Nitric oxide signaling can be altered, affecting vessel dilation capacity. These shifts accumulate gradually, shaped by duration and intensity of activation. The concept of resilience emerges from variability within these systems. Individuals differ in receptor sensitivity. Genetic polymorphisms influence neurotransmitter metabolism. Early life experiences calibrate baseline stress reactivity. Repeated recovery experiences strengthen parasympathetic responsiveness. Biological diversity shapes adaptation patterns. Neuroplasticity ensures that these alterations are not fixed endpoints. Synaptic pruning and growth continue across adulthood. Hippocampal neurogenesis persists at modest levels, provided environmental conditions support regulation. Inflammatory markers decrease when activation subsides. Receptor sensitivity can recalibrate. Chronic stress therefore represents a state of altered regulation rather than permanent damage. Systems operate within modified thresholds. Feedback loops adjust to new set points, yet retain capacity for change. Restoration involves gradual rebalancing of autonomic tone, endocrine rhythms, immune signaling, and neural connectivity. Within this framework, stress becomes a dynamic continuum, not an isolated pathology. Activation and recovery interact continuously. Physiology responding to perceived demands, adjusting moment by moment across endocrine pulses, neuronal firing patterns, and cardiovascular oscillations. And beneath conscious awareness, the organism maintains effort to preserve equilibrium through molecular signaling, receptor modulation, gene transcription, and neural adaptation. The body continuously negotiates between vigilance and restoration, seeking balance within changing internal and external conditions. Even when activation extends beyond its original purpose, regulatory systems remain engaged, adjusting, compensating, recalibrating across time until steadier rhythms gradually regain predominance. As adaptation progresses further, subtle shifts appear in perception itself. Sensory processing may become biased toward threat detection. Auditory and visual systems remain slightly more alert. The thalamus relays signals with heightened priority. Filtering becomes less selective. Neutral stimuli may be evaluated more cautiously. This change reflects protective calibration rather than conscious intention. Within cortical networks, connectivity patterns adjust to repeated activation. Functional imaging studies show altered coupling between limbic structures and executive regions. Stress-associated circuits may dominate resting state activity. Default mode network patterns can shift under sustained cortisol exposure, influencing self-referential thought and rumination tendencies. These neural signatures reflect long-term modulation of network balance. The endocrine system continues its attempt at equilibrium through pulsatile hormone release. Cortisol is secreted in ultradian pulses throughout the day. Chronic stress can alter amplitude and frequency of these pulses. Smaller variations reduce signaling precision. Receptor systems rely on rhythmic fluctuation to maintain sensitivity. Flattened rhythms may impair downstream responsiveness. Thyroid function can also be indirectly influenced by prolonged stress. Elevated cortisol may interfere with conversion of thyroxine into its active form. Subtle shifts in metabolic rate can occur. Energy availability and temperature regulation adjust accordingly. These interactions demonstrate crosstalk between endocrine axes. In reproductive systems, stress signaling may suppress gonadotropin release. This adaptive prioritization conserves energy during perceived threat. Yet when sustained, it alters hormonal balance. Menstrual irregularities or changes in testosterone levels can emerge through hypothalamic modulation, illustrating how survival pathways override non-essential functions during prolonged activation. Within the brain, microglial cells respond to inflammatory signals. Chronic stress may influence their activation state. Microglia participate in synaptic pruning and immune surveillance. Altered activation patterns can modify synaptic architecture over time, linking immune signaling to neuroplasticity. The autonomic nervous system maintains bidirectional communication with the heart through the vagus nerve. Vagal afferents transmit information about visceral state to the brainstem. Chronic stress can reduce vagal tone, diminishing this feedback loop. Reduced vagal input influences emotional regulation and cardiac variability, demonstrating integration between physiological and emotional systems. Metabolic hormones such as leptin and ghrelin interact with stress pathways. Cortisol influences appetite regulation. Reward circuits respond to palatable food with dopaminergic activation. Repeated stress exposure can recalibrate these reward responses, altering motivational salience associated with energy intake. Oxidative stress markers may increase under prolonged cortisol elevation. Mitochondrial function adapts to sustained energy demand. Reactive oxygen species production fluctuates. Antioxidant systems attempt compensation. Cellular resilience depends on balance between production and neutralization. Bone metabolism is also sensitive to endocrine changes. Chronic cortisol elevation can influence osteoblast and osteoclast activity, altering bone remodeling rates. Calcium balance may shift subtly over extended periods, reflecting systemic integration of hormonal signaling. Memory consolidation remains particularly sensitive to stress duration. Acute cortisol may enhance memory encoding for emotionally relevant events, yet chronic exposure can impair hippocampal-dependent recall. Synaptic plasticity mechanisms such as long-term potentiation become less efficient, illustrating the importance of temporal pattern in hormonal signaling. The stress response does not function in isolation from social context. Perception of support or safety modifies amygdala activation patterns. Oxytocin release interacts with stress circuitry, buffering certain physiological responses. Neurochemical modulation demonstrates that stress pathways integrate environmental cues beyond physical threat. Allostatic load accumulates as small deviations compound across systems, not through single catastrophic change, but through incremental shifts in baseline. Blood pressure slightly elevated, sleep slightly fragmented, inflammatory markers slightly increased. cognitive flexibility slightly reduced. These small adjustments form a cumulative profile over time. Yet the capacity for reversal remains embedded within regulatory design. Receptor populations upregulate or downregulate in response to exposure levels. Synaptic strength modifies with altered input frequency. Circadian rhythms can realign when environmental cues stabilize. Immune cells recalibrate sensitivity after prolonged signaling diminishes. Chronic stress, therefore, represents sustained engagement of adaptive systems beyond their intended temporal window. The mechanisms remain fundamentally protective, but their persistence reshapes physiological balance across neural circuits, endocrine rhythms, immune modulation, cardiovascular tone, and metabolic regulation. The organism continues adjusting moment by moment through biochemical cascades and electrical signaling, maintaining function despite altered equilibrium. Resilience resides not in absence of activation, but in the restoration of variability, the return of oscillation, the re-establishment of dynamic range within autonomic, hormonal, and neural systems. And within this ongoing negotiation between demand and recovery, the body preserves its inherent capacity for recalibration. Signaling pathways remain responsive, feedback loops remain operational, adaptation remains possible as long as energy flow, molecular turnover, and neural plasticity continue to support the gradual return toward steadier physiological rhythm. Over extended periods, chronic activation also influences how internal signals are interpreted. Interoceptive pathways transmit information from organs to the brainstem, then onward to cortical regions. When sympathetic tone remains elevated, these signals may be amplified. Normal fluctuations in heart rate or breathing may be registered with greater salience, reinforcing vigilance loops. This amplification reflects altered sensory weighting rather than structural abnormality. The insular cortex plays a role in mapping bodily state. Sustained stress exposure can modify its connectivity with limbic regions, altering integration between sensation and emotional appraisal. Subtle changes in network dynamics influence how internal cues are categorized. Neutral signals may acquire protective significance. Endocrine rhythms beyond cortisol continue adapting. Growth hormone secretion during deep sleep may decline when sleep remains fragmented. Tissue repair processes adjust accordingly. Prolactin and melatonin cycles may shift with circadian disruption, illustrating the interconnected timing of hormonal systems. Blood glucose regulation becomes increasingly dependent on stress hormone modulation. Pancreatic beta cells respond to repeated fluctuations in glucose mobilization. Insulin sensitivity in peripheral tissues adapts gradually. These changes aim to maintain stable energy supply under sustained demand, yet require coordinated feedback to avoid imbalance. The liver participates actively in stress physiology. Cortisol enhances gluconeogenic enzyme expression. Hepatic metabolism shifts toward maintaining circulating fuel. Lipid metabolism patterns adjust in parallel. Triglyceride levels may fluctuate under prolonged hormonal influence. Renal function integrates with sympathetic signaling. Persistent activation influences sodium retention through aldosterone pathways. Fluid balance adjusts subtly, contributing to blood pressure regulation. These interactions demonstrate how multiple endocrine axes converge during chronic stress exposure. Within skeletal muscle, sustained low-level contraction influences metabolic byproducts. Lactate production may increase locally. Muscle spindle sensitivity adapts to prolonged tension. Feedback to the central nervous system reinforces readiness patterns. Neurochemical systems governing mood regulation continue recalibrating. Serotonin transporter expression may change under sustained cortisol. Dopamine receptor density can adapt to repeated reward circuit activation. These modifications occur gradually, reflecting plasticity rather than abrupt shift. Cognitive processing styles may narrow under chronic stress exposure. Attentional bias toward threat-related stimuli increases. Working memory capacity becomes more limited under elevated cortisol. Executive control requires greater effort when autonomic arousal remains high. These functional changes mirror underlying neural network adjustments. The endocrine system's feedback architecture attempts constant correction. Glucocorticoid receptors within hippocampus and hypothalamus monitor circulating cortisol levels. Receptor density adjusts to exposure history. Sensitivity can recover over time when hormonal load decreases, demonstrating reversibility within molecular signaling pathways. Vascular endothelium responds to repeated catecholamine exposure by modifying receptor expression. Smooth muscle tone may shift baseline responsiveness. Nitric oxide synthesis can fluctuate, all reflecting adaptation to sustained sympathetic input. Immune cell trafficking patterns adjust as well. Cortisol influences leukocyte distribution in circulation. Chronic patterns may alter cell migration behavior. Immune surveillance adapts to hormonal context. Oxidative phosphorylation within mitochondria responds dynamically to energy demand. Sustained stress alters mitochondrial biogenesis signals. Cellular respiration efficiency may shift. Reactive oxygen species production requires compensatory antioxidant activity. Cellular systems remain engaged in maintaining balance despite prolonged activation. Epigenetic markers continue mediating long-term adaptation. Histone modification patterns can change in stress-responsive genes, altering transcription rates. These modifications remain potentially reversible, contingent upon environmental input and hormonal context. Throughout all these adjustments, the organism seeks stability through variation. Allostasis depends on flexible oscillation. When oscillation narrows, systems lose adaptive range. Restoration involves widening this range gradually, allowing sympathetic and parasympathetic phases to alternate more freely. The concept of chronic stress therefore encompasses a spectrum of regulatory recalibrations, not a singular pathological state. Each organ system participates in coordinated adaptation. Endocrine pulses, neural firing, immune signaling, metabolic flux, cardiovascular tone, all interacting within integrated networks. Time becomes a central factor. Duration of activation determines depth of adjustment. Short intervals permit full recovery. Repeated cycles without resolution shift baselines, yet even shifted baselines remain dynamic, capable of gradual reorganization when conditions change. Beneath conscious perception, biochemical cascades continue. Receptor binding and unbinding, ion channels opening and closing, second messenger systems amplifying signals, gene transcription responding to hormonal cues, protein synthesis adjusting cellular architecture. These molecular processes operate continuously, shaping systemic physiology over weeks, months, and years. And within this sustained yet adaptable framework, chronic stress unfolds as prolonged engagement of adaptive circuits, each system compensating for persistent demand, maintaining function while gradually redefining equilibrium, until signals of safety and restoration re-emerge strongly enough to recalibrate autonomic tone, endocrine rhythm, immune balance, metabolic regulation, and neural connectivity towards steadier oscillatory patterns once again. As prolonged activation continues across months and years, structural remodeling may extend into connective tissue and extracellular matrix composition. Cortisol influences collagen turnover rates. Fibroblast activity adjusts to hormonal exposure. Subtle changes in tissue elasticity can occur over time, illustrating that stress physiology reaches beyond neural and endocrine systems into structural biology. The hypothalamus remains central in coordinating these responses, integrating input from limbic regions, brainstem nuclei, and circulating hormone levels. Its neurons release peptides in tightly regulated pulses. Chronic stress can alter firing patterns within these nuclei, modifying downstream endocrine rhythms. This adjustment is gradual and reflects persistent signaling rather than sudden change. Within the locus coeruleus, norepinephrine release modulates alertness and attention. Sustained activation enhances baseline firing rate. Cortical arousal remains slightly elevated. Sleep initiation becomes more difficult when this arousal threshold does not fully decline at night. Neurochemical balance between excitation and inhibition shifts subtly under prolonged catecholamine influence. Gamma-aminobutyric acid, the primary inhibitory neurotransmitter, interacts with stress circuitry by dampening excitatory signals. Chronic stress may influence receptor sensitivity within this system, altering inhibitory tone. Balanced excitation and inhibition remain essential for neural stability. The endocrine pancreas responds continuously to fluctuating energy demands. Insulin secretion adapts to repeated glucose mobilization. Glucagon levels shift in coordination. Prolonged imbalance between these hormones can gradually affect metabolic homeostasis, demonstrating interplay between stress pathways and glycemic regulation. In adipose tissue, cortisol influences lipoprotein lipase activity, affecting fat storage patterns. Visceral adipocytes exhibit particular sensitivity to glucocorticoid signaling. Redistribution of energy stores reflects survival prioritization under perceived prolonged demand. The respiratory system also participates. Chronic stress often correlates with altered breathing patterns. Tidal volume may decrease. Respiratory rate may remain mildly elevated. Carbon dioxide levels fluctuate. Chemoreceptors within the medulla respond to these changes, influencing autonomic tone. Baroreceptors located in major blood vessels continuously monitor pressure changes. Sustained sympathetic activation modifies their sensitivity thresholds. Feedback to cardiovascular control centers adjusts accordingly. These micro-adjustments accumulate gradually across time. The endocrine interaction between stress and the renin-angiotensin-aldosterone system further integrates fluid balance with vascular tone. Cortisol can enhance responsiveness within this pathway, linking hormonal stress response to blood pressure regulation mechanisms. In the central nervous system, oligodendrocytes responsible for myelin production may respond to prolonged stress exposure. Myelination patterns influence conduction velocity along neural pathways. Subtle changes in white matter integrity have been observed under extended stress conditions, reflecting long-term neural adaptation. Synaptic pruning processes continue throughout adulthood. Stress hormones modulate expression of neurotrophic factors, such as brain-derived neurotrophic factor. Reduced availability of these growth factors may influence synaptic maintenance, yet levels can recover when stress load diminishes, supporting neural resilience. The circadian pacemaker within the suprachiasmatic nucleus synchronizes peripheral clocks through hormonal and neural signals. Chronic stress can disrupt synchronization. Peripheral tissues may drift slightly from central timing cues. Desynchrony contributes to metabolic and sleep disturbances. Platelets and coagulation factors also respond to stress hormones. Catecholamines influence platelet aggregation potential. Chronic modulation may subtly alter clotting dynamics, illustrating hematologic involvement in prolonged activation states. Within the gut-enteric nervous system communicates bidirectionally with central stress circuits. Stress-induced neurotransmitters alter motility patterns and secretion rates. Microbial metabolites in turn influence neural signaling through vagal pathways, demonstrating reciprocal interaction between microbiota and stress physiology. The endocrine-adipokine interface continues integrating inflammatory signaling. Adipose tissue secretes cytokines and hormones such as leptin. Chronic stress can shift secretion patterns, influencing appetite regulation and immune modulation simultaneously. Neural oscillatory patterns measurable through electroencephalography may change under sustained stress exposure. Beta wave dominance can increase, reflecting heightened cortical arousal. Restoration of alpha and theta rhythms typically accompanies recovery phases, illustrating electrophysiological correlates of autonomic balance. The body, therefore, does not experience chronic stress as a single static state. Rather, it operates within an altered dynamic equilibrium. Feedback loops continue operating. Receptors continue adjusting density. Enzymes regulate hormone metabolism rates. Gene transcription adapts in response to environmental input. Time and variability remain decisive factors. Oscillation between activation and recovery maintains system flexibility. When oscillation narrows, sustained activation predominates, yet flexibility can gradually widen again when conditions permit. Even after prolonged exposure, regulatory systems retain capacity for recalibration. Glucocorticoid receptor expression can normalize. Autonomic tone can regain variability. Inflammatory mediators can decline toward baseline. Neural connectivity can reorganize through plastic processes. Chronic stress thus represents extended engagement of adaptive biological architecture. The same circuits designed for acute survival operate over longer intervals, reshaping physiology through cumulative signaling, yet maintaining intrinsic capacity for adjustment. Beneath conscious awareness, molecular interactions persist, ligand-binding events, phosphorylation cascades, calcium influx through ion channels, mitochondrial ATP production, transcription factor activation, all functioning within living cells, maintaining organismal continuity despite altered demand. And as long as energy substrates remain available, as long as neural circuits preserve plasticity, as long as feedback mechanisms retain responsiveness, the organism continues negotiating equilibrium, gradually modulating sympathetic drive, endocrine pulses, immune signaling, metabolic flux, and cognitive processing toward steadier rhythms when prolonged demand finally subsides. As this extended activation persists across long durations, adaptive recalibration becomes embedded within baseline physiology. Thresholds for response may shift. Smaller triggers initiate measurable autonomic change because neural circuits have adjusted sensitivity through repeated exposure. The amygdala's synaptic strength reflects prior activation history, and cortical inhibitory input may require stronger signals to achieve the same regulatory effect. The brainstem integrates visceral input continuously. Nuclei within the medulla and pons regulate respiration and cardiovascular tone. Chronic sympathetic predominance can subtly influence firing patterns within these centers, altering baseline respiratory variability and heart rate modulation. These changes unfold gradually and often remain below conscious detection. Hormone receptor turnover remains an essential adaptive mechanism. Glucocorticoid receptors undergo cycles of synthesis and degradation. Prolonged cortisol elevation can modify receptor density through transcriptional regulation. Altered receptor availability influences how tissues interpret hormonal signals, creating tissue-specific sensitivity patterns. Within the liver, enzymatic pathways involved in detoxification may respond to sustained hormonal exposure. Cytochrome systems adjust activity based on endocrine signals, influencing metabolism of both endogenous hormones and external compounds. This biochemical adaptability reflects integration between stress physiology and hepatic regulation. Vascular smooth muscle cells adapt to repeated catecholamine exposure by modulating adrenergic receptor expression. Receptor upregulation or downregulation modifies contractile response. These micro-level adjustments shape systemic blood pressure patterns over time. The endocrine axis connecting stress and appetite extends to hypothalamic nuclei controlling energy balance. Neuropeptide Y and pro-opiomelanocortin pathways respond to cortisol signaling, influencing hunger perception and energy expenditure. Sustained modulation alters baseline appetite regulation mechanisms. In skeletal muscle, mitochondrial density may adjust according to energy demand patterns. Chronic stress can influence signaling molecules such as AMP, activated protein kinase, affecting cellular energy sensing. Adaptation seeks to preserve efficient ATP production under altered physiological load. The hippocampus remains particularly sensitive to stress duration. Neurogenesis within the dentate gyrus can be suppressed by prolonged cortisol exposure. Yet when stress load diminishes, neurogenic activity may gradually resume, demonstrating temporal dependency of structural plasticity. Within white matter tracts, axonal conduction depends on myelin integrity. Stress-related hormonal fluctuations can influence oligodendrocyte precursor cell differentiation. Subtle modulation of myelination patterns contributes to altered network connectivity over extended intervals. The endocrine relationship between stress and inflammation involves nuclear factor kappa B signaling pathways. Chronic activation may alter transcription of pro-inflammatory genes. Immune cell receptor expression adjusts accordingly. Immune responses become tuned to persistent hormonal context. Sleep architecture continues interacting with stress biology. Slow-wave sleep supports synaptic homeostasis. Prolonged fragmentation impairs downscaling of synaptic strength accumulated during waking hours. Neural networks remain in a heightened potentiated state, reinforcing vigilance. The cardiovascular baroreflex, responsible for short-term blood pressure regulation, may show reduced sensitivity when sympathetic tone remains dominant. Diminished reflex flexibility influences moment-to-moment cardiovascular adjustments. The endocrine stress response also intersects with bone marrow hematopoiesis. Cortisol influences production and release of immune cells. Chronic signaling can shift leukocyte distribution, integrating stress pathways with immune surveillance capacity. Epigenetic adaptations may extend across generations in some contexts. Parental stress exposure can influence methylation patterns in germ cells. Though mechanisms remain under investigation, this possibility highlights depth of biological integration between environment and physiology. Within cortical circuits, plasticity mechanisms such as long-term depression and potentiation respond to neurotransmitter balance. Chronic catecholamine elevation may bias synaptic plasticity thresholds, affecting learning dynamics and memory encoding efficiency. Metabolic rate fluctuations under sustained stress influence thermoregulation. Sympathetic activation promotes brown adipose tissue activity. Heat production adjusts subtly according to endocrine cues. Energy expenditure patterns reflect integrated hormonal signaling. The organism therefore maintains a layered response structure: molecular changes within cells, network adjustments within brain regions, systemic recalibration across endocrine and autonomic systems, all coordinated through feedback loops designed for stability through change. Time remains the central modulator. Acute stress preserves survival. Chronic activation reshapes regulation. Recovery depends on restoring oscillatory variability across interconnected systems. This restoration unfolds gradually through receptor normalization, synaptic rebalancing, hormonal rhythm realignment, immune recalibration, and metabolic stabilization. Even after prolonged exposure, the architecture of adaptation remains reversible to varying degrees. Plastic processes persist across the lifespan. Cellular turnover continues. Proteins degrade and resynthesize. Neural pathways strengthen or weaken according to input frequency. Chronic stress thus represents sustained engagement of survival circuitry beyond its optimal temporal window. Yet within this extended engagement lies continuous adaptive effort. Regulatory networks do not cease functioning. They adjust thresholds, recalibrate sensitivity, and preserve operational integrity under altered demand. And across neural firing patterns, endocrine pulses, immune mediators, vascular oscillations, and metabolic flux, the organism continues balancing vigilance with the inherent biological drive toward equilibrium, sustaining life through dynamic adaptation, while retaining the capacity for gradual return toward steadier physiological rhythms when prolonged activation eventually recedes. As prolonged adaptation continues, subtle recalibration appears even at the level of cellular signaling cascades. Second, messenger systems, such as cyclic adenosine monophosphate, adjust sensitivity according to receptor activation frequency. Protein kinases modify phosphorylation rates of downstream targets. These biochemical adjustments influence how strongly cells respond to identical hormonal concentrations over time. Ion channel behavior within neurons may also shift under sustained stress exposure. Calcium influx patterns alter intracellular signaling. Gene transcription factors respond to repeated depolarization. Neuronal excitability thresholds recalibrate gradually, affecting how circuits propagate information. Within the endocrine axis, adrenal tissue itself can adapt to chronic stimulation. Enzymatic pathways involved in cortisol synthesis may alter expression levels. Steroidogenic capacity adjusts according to regulatory input from adrenocorticotropic hormone. This feedback maintains output within survivable limits despite prolonged demand. Lipid membranes of cells remain sensitive to hormonal environment. Stress-related oxidative processes can influence membrane fluidity. Receptor mobility within the membrane may shift subtly, altering signal transduction efficiency across many tissues simultaneously. The immune system's communication network of cytokines operates through precise concentration gradients. Chronic stress modifies baseline levels of certain interleukins. These molecules influence not only immune response but also neural activity through blood-brain barrier signaling, illustrating bidirectional integration between immunity and cognition. Vascular endothelial cells exposed to prolonged catecholamines may adjust nitric oxide synthase expression, altering vasodilatory capacity. Microcirculation adapts gradually to persistent autonomic tone. Tissue perfusion patterns shift in subtle but measurable ways. Within the hypothalamus, glial cells contribute to neuroendocrine regulation. Astrocytes modulate neurotransmitter clearance and metabolic support. Chronic stress can influence glial signaling pathways, affecting synaptic microenvironments and neurovascular coupling. Metabolic flexibility depends on the body's ability to switch between glucose and fatty acid utilization. Sustained cortisol exposure influences enzymes involved in beta oxidation. Cellular fuel preference may adapt to repeated stress signaling, reflecting coordination between endocrine and mitochondrial regulation. The renal system integrates stress hormones into filtration dynamics. Glomerular blood flow responds to sympathetic tone. Tubular reabsorption processes adjust under aldosterone influence. Fluid and electrolyte balance remain tightly regulated despite ongoing activation. Within skeletal tissue, osteocytes sense mechanical load and hormonal signals. Chronic glucocorticoid presence may influence their signaling to osteoblasts and osteoclasts. Bone remodeling equilibrium shifts gradually under sustained endocrine modulation. Neural connectivity within large-scale brain networks continues adapting. Functional connectivity between salience network and executive control network may strengthen under prolonged stress exposure. This shift biases attention toward potential threat detection, while reducing spontaneous exploratory cognition. The default mode network, involved in self-referential processing, can exhibit altered coherence when cortisol rhythms remain dysregulated. Persistent activation influences internal narrative processing patterns, reflecting integrated neuroendocrine modulation of cognition. The microbiota gut-brain axis further demonstrates chronic stress integration. Microbial diversity may decrease under sustained autonomic imbalance. Microbial metabolites influence vagal afferent signaling. Short-chain fatty acids interact with immune pathways, linking digestive ecology to neural regulation. Within hematologic systems, red blood cell distribution and plasma viscosity may adjust in response to sustained sympathetic signaling. These parameters influence oxygen delivery efficiency, reflecting systemic integration of cardiovascular and metabolic adaptation. Thermoregulatory centers in the hypothalamus respond to stress hormones by modulating peripheral vasoconstriction and sweat gland activity. Chronic sympathetic tone may alter baseline thermoregulatory thresholds, producing subtle shifts in perceived temperature stability. Neurotransmitter receptor internalization cycles continue responding to exposure patterns. Prolonged catecholamine release can lead to receptor desensitization, followed by compensatory upregulation during recovery. These dynamic adjustments preserve signaling balance over time. The endocrine interplay between stress and circadian timing influences peripheral clock genes. Transcription factors within liver, heart, and adipose tissue oscillate according to hormonal cues. Chronic disruption can dampen amplitude of these oscillations, reducing temporal precision of metabolic regulation. Over extended durations, neural dendritic architecture may remodel in region-specific patterns. Stress-sensitive areas such as hippocampus and prefrontal cortex show structural plasticity in response to hormonal load, while amygdala circuits may strengthen synaptic connectivity. These regionally distinct adaptations reflect functional prioritization under sustained vigilance. Yet despite the breadth of physiological influence, the organism maintains core homeostatic parameters within survivable range: pH balance, oxygen saturation, electrolyte concentration, and core temperature remain tightly regulated, demonstrating resilience of fundamental life support systems even under chronic activation. Restoration remains dependent on reintroducing variability into autonomic cycles. Parasympathetic tone gradually reasserts influence when environmental cues shift. Hormonal pulses regain amplitude. Inflammatory mediators decline toward baseline. Receptor sensitivity normalizes through turnover and gene regulation. Plasticity persists across molecular, cellular, and systemic scales. Neurons continue forming and pruning synapses. Endocrine glands adjust secretion rates. Immune cells recalibrate responsiveness. Metabolic enzymes regulate substrate flux. Chronic stress, therefore, represents extended modulation of adaptive networks rather than irreversible collapse. The body preserves operational integrity through layered compensatory mechanisms, maintaining coherence across endocrine, neural, cardiovascular, immune, and metabolic systems. And through continuous feedback, receptor adaptation, gene transcription modulation, enzymatic recalibration, and synaptic restructuring, the organism remains engaged in dynamic negotiation between sustained demand and inherent biological drive toward equilibrium, preserving the possibility of gradual return to broader oscillatory balance when prolonged activation eventually diminishes. Over long durations, chronic stress becomes less about intensity and more about persistence. Activation that once served immediate survival reshapes baseline regulation through repeated signaling. Hormones continue circulating in altered rhythms. Neural circuits maintain adjusted thresholds. Immune mediators sustain modified patterns. Cardiovascular tone remains subtly shifted, yet none of these changes occur in isolation. Each adjustment reflects coordination across interconnected systems seeking stability under sustained demand. The stress response was never designed to remain constantly engaged. It evolved to rise, resolve, and return to baseline. When resolution does not arrive, feedback mechanisms compensate as best they can. Receptors recalibrate, enzymes adjust synthesis rates, synapses reorganize, inflammatory pathways adapt sensitivity. The organism preserves function even while equilibrium shifts. Within this extended adaptation, the principle of allostasis remains central. Stability is maintained through change, but change carries energetic cost. Cumulative signaling gradually narrows variability. Heart rate variability declines. Hormonal amplitude flattens. Sleep cycles fragment. Cognitive flexibility reduces. These subtle contractions of dynamic range define the physiological footprint of chronic stress. Yet biological systems retain inherent plasticity. Receptor populations turn over. Gene expression patterns respond to new environmental input. Synaptic strength modifies with altered signaling frequency. Mitochondrial efficiency adapts to shifting energy demand. Immune cells recalibrate receptor density. These processes demonstrate that regulation is dynamic rather than fixed. The organism continuously negotiates between vigilance and restoration, sympathetic activation and parasympathetic recovery, cortisol secretion and negative feedback inhibition, inflammatory signaling and anti-inflammatory modulation. Oscillation between these poles determines resilience. When variability widens once more, rhythms begin to normalize. Circadian patterns regain amplitude. Sleep architecture deepens. Vagal tone increases. Inflammatory markers decline. Cognitive processing regains flexibility. These shifts occur gradually through molecular turnover and network rebalancing. Chronic stress, therefore, represents prolonged engagement of adaptive survival architecture, not a singular malfunction, but a sustained state of recalibration across endocrine, neural, immune, metabolic, and cardiovascular domains, each system adjusting thresholds to maintain continuity of life under repeated demand. And beneath conscious awareness, countless molecular interactions continue. Hormones binding receptors, ion channels opening and closing, transcription factors regulating genes, mitochondria generating energy, neurons firing in coordinated networks, all working within the living organism to preserve coherence. As long as energy supply persists, as long as neural plasticity remains intact, as long as feedback loops retain responsiveness, the potential for re-expansion of physiological variability remains present. Equilibrium is not a fixed point, but a dynamic range. And within that range, the body continuously adjusts, recalibrates, and sustains itself, even after prolonged activation, guided by the same regulatory principles that have supported survival across evolutionary time. If you enjoy peaceful stories like this, you are warmly invited to subscribe and return whenever you need to slow down.